Hello everyone, welcome once again. Today we are going to talk about a topic in forensic science titled Collection and Preservation of DNA Samples. So, we are going to look at what happens when we get to a biological crime scene. So, when we get to a biological crime scene, how do we collect and preserve biological or DNA evidence? Also, at the forensic lab, when suspects are brought for sample collection, how do we collect such samples? At the site of disasters, or even at the mortuary during autopsies, if we want to pick samples from bodies for DNA testing, how do we pick and preserve these samples? That is what we are going to talk about today. So, these are objectives for this discussion. At the end of the day, you have a general understanding of what DNA is. We also get to know where DNA can be found. Then, we also get to know the use of DNA in forensic science. And we will know the meaning of two terms, evidence and reference DNA samples. Then, we will know how to collect evidence and reference DNA samples. And we also know how to properly store and preserve DNA samples. So with introduction, we say DNA technology is a new tool in forensic science. If you look at the various forensic disciplines, DNA is quite new. So it's a new tool in forensic science. And it has also replaced blood grouping in most forensic laboratories. Before DNA came, forensic scientists were using blood group to link perpetrators to crime scenes. But then DNA has come to replace blood group in many forensic labs. Though blood grouping can still be done in some ways. But then because DNA is unique to individuals, so most forensic laboratories prefer DNA testing to blood grouping. As in all cases of science, evidence from forensic DNA investigation does not mean proof. It means if DNA from the crime scene matches with DNA from a suspect or victim, especially the suspect, it doesn't mean or it doesn't prove that the suspect committed the crime. But in forensic science, what we can say is that the sample taken from the suspect matches with the sample that was taken from the crime scene. Whether the person committed the crime or not, that is up to the court of law to determine. So what is DNA? It's an acronym for deoxyribonucleic acid. And we have two types. We have the nuclear DNA, and we also have mitochondrial DNA. The nuclear DNA is obtained from the nucleus of a cell. The mitochondrial DNA is obtained from an organelle called mitochondria. And in terms of how we inherit them, we inherit nuclear DNA from both parents, that's the father and the mother. they all contribute to the nuclear DNA of a child. But then when we talk about mitochondrial DNA, only mothers contribute to this type of DNA. So when you pick the mitochondrial DNA of a child, it matches with that of his or her mother. Nuclear DNA, as we've already said, is inherited from both parents, father and mother. And it is found in the nucleus of all cells except. Can you help me answer this? We say nuclear DNA is found in the nucleus of all cells except there was one particular cell that doesn't have this nuclear DNA. So, this particular type of DNA cannot be found in matured red blood cells. Why? Because matured red blood cells do not have a nucleus. And once they do not have a nucleus, then it automatically means that they cannot have nuclear DNA. And we say nuclear DNA is unique for an individual, except monozygotic twins, that's identical twins. They have the same nuclear DNA because they were formed from the same egg and also the same sperm. So they are the same in terms of genetics. And nuclear DNA is the most common DNA used in forensics, biotechnology, diagnosis, etc. But nuclear DNA is prone to degradation. And there's a question here, why? I hope you've answered this question. This is because when you pick a cell, there is only one nucleus. But then if you look at the mitochondria, there are several mitochondria within a cell. So when there is degradation, you are likely to lose almost all nuclear DNA. But because there are a lot of mitochondria in a single cell, you still have some mitochondrial DNA intact. Now let's move to the second type of DNA, which is the mitochondrial DNA. And we say this form of DNA is inherited from the mother alone, or the mother only. Why? I hope you've answered this. Mitochondrial DNA is inherited from the mother alone because during conception, it is only the mother that contributes her mitochondrial DNA to the fetus. Once the head of a sperm enters an egg, fertilization takes place. And normally the mitochondrial DNA of a sperm is found in the tail. And since the tail is left out, it means the father is going to contribute only nuclear DNA to the fetus. That is why we say it is only the mother that contributes her mitochondrial DNA to the child. And mitochondrial DNA, as we've already talked about, is found in mitochondria, which is an organelle in the cell. Another unique feature of mitochondrial DNA is that all maternally related individuals have the same mitochondrial DNA. So each and everyone from the same maternal line have the same mitochondrial DNA. This makes it useful in lineage studies. And also, mitochondrial DNA It's not very useful in criminal investigations. Once again, a question has been posed. Why? Why is mitochondrial DNA not very useful in criminal investigations? The answer is simple. In criminal investigation, when we are using DNA technology, we always want to link or pinpoint a crime scene evidence to an individual or a suspect or perpetrator. And this is the case that we've already said, that mitochondrial DNA is the same for each and everyone from the same maternal line. What does it mean? It means if you use mitochondrial DNA in criminal investigation, you can't pinpoint the crime scene evidence to a single individual. Why? Because there's a possibility that the individual that we are pinpointing it to, they didn't leave the sample at the crime scene, but then another person coming from the same maternal line as our suspect. So now our suspect become each and every one from the same maternal line as our initial suspect, and not just a single person. So when you're talking about criminal investigation, we normally prefer nuclear DNA and not mitochondrial DNA because it is not unique. But then it's very useful when nuclear DNA is degraded. You can resort to mitochondrial DNA, especially in missing person identification. When are we likely to find DNA evidence at a scene of crime? In homicide cases, in sexual assault cases like semen, burglary and break-ins, maybe blood, and other cases, a whole lot of cases, we can find DNA evidence. Where can we find DNA? We can find DNA in almost every biological sample: blood, semen, saliva, bone, teeth, nails, being fingernails or toenails, and hair. But then there's a question mark there. If you want to use hair in DNA testing and you want nuclear DNA, the hair must possess the root because the shaft contains mitochondrial DNA but not nuclear DNA. Uses of DNA. How do we use DNA in forensic science? 1. We use DNA evidence to link suspect or victim to a crime scene. 2. To link suspect to victim and vice versa. 3. We can use DNA evidence to connect two or more crime scenes. 4. We can use DNA evidence in the identification of a missing person. We can also use DNA evidence in disaster victim identification. Also, we can use DNA in relationship testing, the maternity testing, the paternity testing, siblingship testings, etc. Now let's look at some DNA collection case. If you want to collect DNA evidence either at the crime scene or from suspects, what are the case or the necessary instrument that you need? 1. We need a sterile swab. Sterile swab means this swab is devoid of any contaminant. be it foreign DNA or microorganism. We also need paper envelopes. We need DNase-free water. DNase is an enzyme that can degrade DNA. So the water we are going to use should be devoid or free from this particular enzyme, because this enzyme can degrade the DNA we are going to take. Then in some cases, we might need surgical blades. These are a few of the collection kits. Now, processing DNA evidence at crime scenes. What steps should you follow in order to locate, collect, and preserve DNA evidence when you find yourself at a scene of crime? First, your health is paramount. So you have to wear your protective clothing like Tyvek and overall clothing. 2, you then have to locate and mark all your evidence properly. Then you can photograph all your evidence. After photography, you can then collect all evidence and package them separately. You don't package biological evidence together. You package them separately. Then labeling. This is very important. You have to label all your biological evidence very well. During collection, you have to change gloves for every evidence collected. And why do you think it is necessary for a crime scene investigator or biological evidence collector to change gloves for every evidence that he or she is going to collect? The answer is simple. If you don't change gloves, there is a high possibility that you transfer evidence that you initially pick to the next one. Because once you use a glove to pick an evidence, special biological evidence, it can get stained into the glove. So if you don't change the glove and you use the same glove in picking another biological evidence, you are going to transfer to that one. That is why we need to be changing gloves from one evidence to the other when we find ourselves at a biological crime scene. And also, during collection of biological evidence at a scene of crime or even at the laboratory, we must try to avoid talking as much as possible. Once again, why? The answer over here is also quite simple. As we talk or express ourselves, we do bring out saliva, which is also a form of biological material and contains our DNA. So, if you talk or you keep on talking during collection of biological evidence, there is that likelihood that you transfer your own biological evidence, in this case your saliva, to the evidence you are going to take, which will result in contamination. I hope you've understood the reason or the answer to this question. Then you transport biological evidence as quickly as possible to the forensic laboratory. you don't delay, or you have yourself to blame, because you can end up destroying this biological evidence. Now, proper evidence collection is aimed at avoiding contamination, ensuring safety and health of personnel, and also preservation of the evidence. Collection, preservation, and packaging. We say collect DNA evidence as quickly as possible. Why? Because DNA evidence are normally prone to degradation. That is why we have to collect them as quickly as possible to prevent them from going bad. Now, the first biological evidence that we are going to talk about, body fluids. These are fluids from any part of our body. That biological evidence that is fluid or liquid in nature can be termed as body fluid. How do you collect such evidence? We say collect dried body fluids with a wet swab. It means if you get to a crime scene, you find biological evidence or body fluid, but has dried up. You have to wet a sterile swab before you can pick such a sample. So use DNA-free water to moisten the swab. Why? We've already explained this, so answer it for yourself. Collect wet stains with a dry swab. If you come across a body fluid which is still in its original state, liquid, you just have to dip your sterile swab into it. By doing so, you've collected the biological stain. Then you collect 2 or more swabs for a single stain. Don't collect just a single swab. Collect 2 or more from a single stain, so that should something happen, the forensic science lab can get other sample for retesting. Then all swabs must be air dried before packaging. Why do we have to air dry it and not in the sun or using an oven, special microwave oven? Heat can also destroy or degrade DNA. So drying body fluid in the sun or using the heat of a microwave isn't advisable. We always try to air dry it using room temperature. Now all swabs must be packaged separately. Why? This is because the evidence that we've picked at the crime scene might be coming from different sources. So once you pick stain A, you pick stain B, and you package them together, there will be that transfer of material leading to contamination. And if, once again, if these 2, 3, or more swabs came from different individuals, it means you are going to have mixed DNA profiles after a forensic lab process this type of evidence. So in order to avoid this, always package each and every evidence separately in different paper bags or paper envelopes. Then we say collect whole blood in vacutainers with anticoagulant and keep frozen. If you have whole blood, you preserve them in vacutainers with anticoagulants such as EDTA, after which you can keep them frozen. Or you can even take swabs of this whole blood, air dry it, package it in paper envelopes, and then you store it at room temperature. You also collect and package clothes and beddings with stains if possible, or cut the area with the stain. If you come across clothes or beddings that has been stained with suspected body fluids, you can either cut those portions with the stains, or you can package the entire clothes of beddings and transport them to the forensic laboratory. But before packaging, make sure the stains have been air dried. Now, all samples after collection must be packaged in paper envelopes and transported to the lab in dry state. Or, if the sample is in its liquid form, example, whole blood in vacutainer tubes, you transport them on ice. Air drying and paper envelopes preserve DNA evidence prior to grant transportation to the forensic lab. Knives with stains can be packaged wholly in cardboard boxes. It is not advisable to package knives in paper envelopes because these are sharp objects. So you always package them in cardboard boxes. Now let's move on to the second type of exhibit or sample that we can come across. These are hard tissues, bone, teeth. How do we collect, package, and transport them? We say collect and package once again in paper envelopes. You then transport in dry state or on ice. You can also use liquid nitrogen to preserve this type of evidence. So you put them in liquid nitrogen. Nails, either fingernails or toenails, you air dry and package in paper envelopes. If you pick fingernail scrapings in sexual assault cases, you must air dry it and also package in paper envelopes. During sexual assault, you are likely to get biological material from the victim or from the perpetrator in the fingernail of either the victim or the perpetrator. So let's say you find biological material that you are suspecting to have come from a perpetrator, sexual assault perpetrator, in the fingernail of a victim. You can use a wet or moistened swab to pick this material inside the fingernail of the victim. You then air dry it and package in paper envelope for transportation. Hey, use tweezers to collect hair evidence for DNA. If this hair contains the root, don't break the root. Because if your interest is nuclear DNA, it is the root that contains the nuclear DNA. So make sure the root is intact. Then use different tweezers to collect each hair to avoid contamination. Or if you don't have a lot of tweezers, decontaminate the tweezers thoroughly before further use, using 5% bleach, after which you redecontaminate with 70% ethanol to remove any excess bleach. Muscle or soft tissue. Collect muscle in appropriate tube and transport on ice to the lab or keep frozen to preserve the DNA. So you can either collect it in a sterile tube or even in EDTA vacutainers and transport it on ice to the lab. Touch DNA. What is touch DNA? Based on locals exchange principle, every contact leaves a trace. It means once we touch something, we leave our biological material there. So if you want to enter a building, once you hold the doorknob, you've left your biological material. Fingerprint, which also contains DNA, though the success rate of such DNA is very low. But it is still a good source of biological material. So how do we collect touch DNA? We swab the suspected area or item. We then air dry the swab and package in paper envelope for transportation to the lab. We've already talked about evidence sample. All this while, we are talking about evidence samples. That is sample that we get from a scene of crime because we don't know the source. The source of such samples are unknown. So, if we get such samples, it means we need something to compare because forensic science is all about identification and comparison. You identify something, you then need another thing to compare whether they match or not. That is forensic science. So, after working or picking these evidence samples or samples of unknown sources, we need known source or standards to compare. Where do we get these reference materials? 1. From our suspects or victims. 2. We get them from either antemortem or postmortem samples. In some cases, we can use close relatives for identification. What type of samples do we normally take for comparison purposes? Buccal swabs or cheek swabs from suspects or victims using sterile swabs. Buccal swabs from relatives, close ones, are preferable. Antemortem samples, biopsy samples from razor blades, toothbrushes, hairbrushes. Postmortem samples, blood without formalin, because formalin can interfere with DNA analysis. Also nails, either fingernails or toenails, and then soft tissue muscle. All buccal swabs must be air dried, packaged in paper envelopes, and stored at room temperature. All evidence and reference DNA samples must be transported as quickly as possible to the forensic lab. Proper documentation should be attached to every sample submitted to the lab. So, we are now going to do a lot of discussion. In our next slides, we are going to look at some pictures. And then, at our individual places, we try to figure out if you happen to find yourself at such a scene, what evidence in this picture or pictures are you going to pick for DNA testing? So this is the first picture, sorry. What do you see here? And which one do you think will be very good for biological sample collection? I can see a fruit juice container lying there. And since someone might have drank it, we can swab it for possible saliva. Then, can also see a clothing here. We can analyze it for the presence of body fluid. You can continue with the rest because I can even see an empty soda can here. Next picture. I hope you've been able to pick some important items out. So let's move on to the next picture. The next picture. The next one. So I hope we're able to pick some items for biological sampling. Conclusion. We say DNA evidence, if properly collected, preserved, packaged, and transported, can be very useful to the criminal justice system and also in all other forms of human identification. Thank you very much for your audience. Once again, please don't forget to like, share, leave your comments, and also subscribe to this channel for more exciting presentations. Thank you once again for your time and do have a good day. Thank you.